All right, I'm going to go ahead and get started just in the interest of time, because I want to be sure that we have plenty of time to have the opportunity for students to join the researchers in the breakout rooms. I think that's probably the most valuable part of this. So first of all, hi, I am Dr. Vicki Channon. I am a teaching assistant professor in psychology and neuroscience, and I serve the role of our director of undergrad research for psychology. And so if you have any questions about how to get involved in research or anything, definitely feel free to reach out. Make sure you check out the Sakai site. There are some tips in getting involved and everything like that. But so for today's session, we have 5 researchers that are going to join us to talk about research happening in their lab. The format really is just meant for them to give kind of a brief overview. I have it set up so presenters, you are able to share slides if you want. Try to be mindful of time, just so that, again, we have plenty of time at the end for students to actually have the chance to talk to you. If you go much over 5 minutes, I might start to butt in a little bit just to remind you to try to finish it up. Again, because I think that the more people they're able to talk to then, the better it will be. So I'm going to go ahead and get started. And this is the order that we'll go in. And again, each researcher will speak briefly, and then I'll open up 5 breakout rooms that will be numbered in the same way that you see on the screen. And I can bring this slide back up later as well. Let me do that. Okay, so I am going to stop sharing. Okay. So the first person, and now that the slide went away, I don't have it up here anymore, but the first person that we're going to hear from is Claire Mink. And so Claire, I will go ahead and turn it over to you.
Sure, thank you. I did throw together a couple slides, so I'll share them, but I'm also, I'll be quick. If I can get it to correct. My name is Claire. I am a senior actually, and I'm an undergraduate. I'm doing an honors.
Can other people hear Claire?
Can you hear me?
Okay, it's just me.
Okay, everybody can hear me? Okay, lovely. So my PI couldn't be here today, so she asked me to kind of present to you guys, but I am a senior psychology major. I'm an undergraduate, and I'm doing an honors thesis in the lab. So that's kind of my role. Just really quickly about me. I was like a pretty new psychology major. I declared like my sophomore spring. So I've crammed a lot into a short amount of time, but I did 395 last semester, and now I'm doing my honors thesis. So I'm primarily interested in clinical and social psychology, which is what I've ended up being able to do with Dr. Husong. So like I said, Dr. Andrea Husong is the PI for our lab. Primarily, our lab is doing research on alcohol use as well as gratitude in children. And then we have a couple of projects going on examining like social media habits, which I think is really interesting. Here's a quick list of some of the kind of past, like recent projects or like projects in progress that we have going on in the lab that are really interesting, and I'd love to talk to you guys about. And then just lastly about like what I specifically do, my thesis is examining the relationship between depression and alcohol use by using a text analysis of like text messages, like between phones and stuff. So I'm using like a secondary analysis from a study that we did in the lab. And I'm excited to talk about it more with you guys. So that is all.
Awesome, thank you. Yes, so you got, I think, kind of an overview of the things that they do in Claire's lab that you could talk with her about if you decide to join her breakout room. But the other thing is, if any of you are wanting to know more about how to do an honors thesis or anything like that, that might also be a good breakout room to join to ask about that. Okay, so our next presenter will be Guaran. Is that how you pronounce your name? I'm sorry.
Yes, right, it's correct. Perfect.
Thank you.
Thanks, Vicky, to organizing this. And hi everyone. I'm the PI of the computational lab in computer science and psychiatry. So basically, I'm I'm a computer scientist, and but I'm interested in understand the human brain. So our lab has three focus. So first is we develop the computational tools to process the raw image. So basically, we will turn the raw image into a data structure that the computer can recognize. So we have the pipeline to process the structure image, diffusion tensor image, and the functional image. And we have the pipeline. So if you want to have the skill set to process the image, so I'm sure there's a lot of projects that you can work with us. And with these tools in place, and we're also interested to use these tools to find the biomarkers and to do the early diagnosis of the neurological disease. So currently we have the project for the human and also for the animal model. So for the animal model, we are specifically interested in understanding the autism. So we're working with the new couple labs at the Neuroscience Center. So we use tissue clearing and microscope imaging technology to understand how the genetics will affect the brain structure. And so I think the overarching goal is to understand the mechanism of autism, whether it's it's something in the genetics or something in the neurogenesis. Okay, I'm not an expert, and I don't have the training by neuroscience, but my role in this project is to develop the computational tools to analyze the larger scale microscope image, to count the number of the nuclei and to analyze the shape and to see how they related to the disease. And we also have the study for the human and with focus on in Alzheimer's disease. So this is a recent project. We want to understand why we in the clinic we see more like a female has high risk to develop Alzheimer's disease than males. And we want to see whether this difference, sex and gender difference is something to do with biology or something to do with environment. So we'll use our computational method, which including machine learning, statistic analysis. We try to analyze the role of the interaction between gene and environment. And so the last part is very fun. So it's about information geometry. So basically, we are working with the mathematician and the statistician. We try to understand, find the geometric patterns from our neuroimaging data. So let me use a quick video to show what I mean by geometry. So the overarching goal is to understand the dynamic of functional, our brain functions. So how the structure support the function and how the function emerge the behavior. So it's very hard to understand dynamics. So it's kind of like there's a lot of birds, they're flying and they form some dynamic patterns. We try to get some using the mathematics tools to get to characterize this oscillation pattern. So this is one of our research focus, and another is we try to so this is kind of like how to understand our cognition. So I use this example. So let's say there's ants, right? So let's suppose this ant is living in a 2D world, and this ant only know to walk from the left to the right and up down. Okay, it's complete on 2D dimension. But if we can create another dimension, we can fold this space, right? So and that probably can answer the question that why we in our human brain we see the brain that's supposed to connect by the white matter fibers, but why sometimes the region is they are far apart, but they can quickly connect it, and we don't understand why. We don't have the mathematic tools to answer this question. So this is something that we need a new mathematics framework or theory to answer this question. So I think that's also, if you need, if you want to learn more about my lab, just come to talk to me. All right, thank you.
Perfect. Thank you so much. Y'all are doing a much better job at keeping to the time limit compared to how it went yesterday. Okay, wonderful. So next up we have Shelby Coleman.
Hi, everybody. We actually don't have slides, so we are just going to chat with you all today. So I actually have Rachel Namira here, who is one of our undergraduate research assistants working in the CLIC lab. So I'm going to let her actually talk for most of the time about her experience working with CLIC, but I did just want to introduce myself, the PIs in the lab, and Rachel a little bit. So I am Shelby Coleman. I am a postdoctoral research associate in the CLIC lab. So I got my PhD from the University of Georgia. And I work with Dr. Jeff Green and Dr. Matthew Bernacki in the School of Education in the CLIC lab, and they are the main PIs on the projects that we do in the CLIC lab. And then we love working with undergraduate students. We've worked with undergraduate students in both the School of Education and the Psychology department, which is actually where Rachel is coming from. So I'm really going to let her tell you more about kind of our research and what she has been doing in the lab, but I will be hanging on after for breakout rooms. So super excited to chat with anyone who might be interested in our research. So Rachel, I'll let you take it away.
Thanks, Shelby. So I'm Rachel Namira. I'm a junior here at Chapel Hill. I'm a double major in psychology and anthropology, and I just declared a minor in cognitive science as well. I'm also a transfer student if anybody has any specific questions related to that. And this is my second semester working as an undergraduate research assistant with the CLIC lab. So CLIC stands for Critique, Learn, Investigate, and Construct Knowledge, which is what CLIC studies in educational contexts. Specifically, we conduct research on how students self-regulate their learning, which are the iterative processes through which students monitor and control their cognition, motivation, and behaviors during learning. Many members of our lab study how students self-regulate their learning in technology-based learning environments, because these environments are often complex and contain a lot of information, which can be really demanding for students and can require effective self-regulation to navigate. Some members of our lab also study epistemic cognition, which is the process through which people construct, evaluate, and use information. Epistemic cognition is also a necessary skill for learning successfully in our current world where we have so much constant access to information. The CLIC research group is in the School of Education, but we work with students and faculty in the psychology department, as Shelby was saying, and also other departments across the university. So about me personally, I had always been interested in doing research at Carolina, so I had my eye open for positions at psych labs. And I actually found out about the CLIC lab through like an email from the psychology listserv. So I looked up the lab and was super excited to find out that they were researching all of the topics that I was really interested in. So I applied through the online form and the email, and then later I had a Zoom interview with some of the doctoral students and with Shelby. So last semester I worked in the lab through 395, like Claire said earlier, and this semester it's been serving as my work study position through the university. Within CLIC, what I actually do is a variety of things. Last semester, most of my work focused on remote data collection over Zoom. So I would meet with participants and guide them through a process. And I also did some data analysis and coding. This semester, as I've learned more and I've become more comfortable with the content of the lab, I've been able to take on a little more responsibility. So I recently got the opportunity to contribute to a paper that will be submitted for publication in the fall semester. And I've been diving into more of the literature on self-regulated learning, and soon I'll be taking the lead on coding a large set of intervention data, which I'm really excited about. Overall, I can't say enough positive things about my time with the CLIC lab. It's been extremely rewarding. Something that I didn't realize how helpful it would be is that I have been attending weekly meetings with the graduate students, postdocs, and professors. And this is giving me like a really unique look into the kind of work that I could be doing after my undergraduate career. Which has been extremely exciting. I've also gained more experience in data collection and analysis, and I've worked on my writing skills to be more professional and academic. So yeah, overall, I can't recommend getting into research enough, and I'm excited to help you guys find similar opportunities.
Thank you so much. Yes, and I really like that Rachel commented on one of the benefits really being getting to like know a bunch of grad students and postdocs and kind of seeing how the whole game, right, like of academia really works. Like in some cases, I think that's like getting to know those people is even more beneficial than like learning the research skills. I know I would have been totally clueless headed into grad school if I didn't do research as an undergrad. All right. Next up is Dan Christoffel. I believe that's how to pronounce your name. I'm not sure.
Yep, that's correct. Thank you. A lot of people get it wrong, so thank you.
I remember that from the lab meeting and I caught it and remembered. Or from the faculty meeting.
Yes, that's right. All right, so I'll share, I have a few slides and just give you guys a brief overview of what we do in the lab. So we all good? Can everyone see it? Yes, okay, great. So I'm a new PI in the Behavioral and Integrative Neuroscience program in the Department of Psychology and Neuroscience. And I use rodent models to investigate how the brain regulates and processes rewarding experiences and how this can sometimes go awry in things like stress, depression, overeating behaviors. And currently my work's focusing on palatable food consumption. And so you can see the title of Neurobiology of Allostasis is basically just the study of how the brain adapts to change and experiences over time. So this is what my lab does and uses modern neuroscience tools to begin to investigate this question. Another way of saying that is another fancy phrase, which is experience-dependent plasticity, which is again just really a way of saying how does the brain learn and how does the brain change when it learns an experience or it goes through multiple experiences. So I study palatable food intake and fooding behavior. So that's one level of what the lab does. So we give animals either limited or chronic access to high-fat food versus sort of normal, standard, healthy food. And we analyze their feeding behavior sometimes for an hour. And we do this with some advanced machine learning video analysis software that allows us to automatically detect when the animal's approaching the food, when it's eating, so we can sort of get a very detailed look at the behavior as opposed to just looking at they ate, X calories of food in that time. And then once we can do that, we want to manipulate the brain and observe the brain to see how we can change the behavior or what's going on in specific brain regions during the behavior. So that's sort of the neural circuit level. We use neuromodulation tools that are called optogenetics and chemogenetics that allow us to either turn up or on the activity of certain cells in the brain or turn them off. And then we use another technique that's called calcium imaging based photometry, which allows us to observe the activity of specific cells in the brain and link that activity to the behavior that we've gained from the video analysis portion. And then finally, we try to take it down to the point of connection between cells or neurons in the brain and looking at synapses. And we do this by measuring electrophysiology and acute brain slices that we remove and keep alive for a day. And then we're able to measure the electrical activity and the release of neurotransmitters. And then we also stain for different proteins and do imaging to sort of analyze if the abundance or location of proteins shift over time, given, a particular manipulation or given a particular experience, for example, chronic high fat intake versus a normal healthy diet. And so that's sort of the broad overview, but basically the sort of pitch line is if you're interested in investigating how much of a good thing alters the neural circuits and affects behavior, then you might be interested in our research, focusing on looking at hedonic feeding. And then we're also looking at the roles of the neuromodulators, dopamine and serotonin on feeding behavior. And I have my lab website if you want to go take a look and learn a little bit more about the research I've done. And this is the lab team thus far. So it's myself, a technician who earned her master's just this past year at the University of Buffalo, and a research volunteer who comes from UNC Charlotte, who's starting to learn how to do rodent research. And so these two individuals should give you an idea of what's going on in the lab. So that's it. Look forward to talking to you all later.
Great, thanks so much. All right, and then last but not least, we have Lisa Tarantino is going to tell us about.
Okay, so you saved the best for last. All right, so my, hi everyone. So my laboratory is in the Department of Genetics, and we study behavioral genetics. We're primarily interested in addiction, and like Dan and others, we use animal models, so primarily mouse models. We're located in the Genetic Medicine Building, which is over across Manning Drive, about as far north on campus as you can get. So it's pretty clear from human studies that the risk for addiction is fairly high and genetically controlled. So things like cocaine and opiates have a heritability of about 0.7, which is fairly high. And so we are interested in identifying genes that increase risk for substance use disorders, and in my lab, primarily cocaine, but we've also done work with opiates and other psychostimulants as well. So as I said, we use mouse models. We spend a lot of time working with the collaborative cross mice, which are located here at UNC. They are an 8-way cross between 8 different inbred mouse strains, and they were crossed together to create this heterogeneous population. Each mouse within a collaborative cross strain is genetically identical to other mice within that strain, so they're inbred strains, and there's about 75 different strains, so there's a lot of genetic diversity in this population. So what we do is study naturally occurring phenotypic variation on this controlled genetic background, and then use that to perform systems genetic studies. So our lab is, it's difficult to study addiction genetics in humans because of issues with environmental influences and the drug use histories in humans that present at treatment clinics. And so we are using mice to kind of be able to have some better control over drug exposure and to be able to study the effects of drugs from the first use and then into more repeated exposures and hopefully addiction behaviors. So some of the behaviors we do in the lab are look at initial drug sensitivity, which is a locomotor response to an initial dose of a drug, in this case cocaine. We do a three days experiment where we inject mice with saline on days 1 and 2. We put them in this open field here, which is essentially just a big box that measures their locomotor activity and where they are. And you can see that down here, that when an animal is given saline, these red lines show where the animal is traveling in this open field. When you give them cocaine, you can obviously see a lot more red because the mice are running around a lot more, because obviously cocaine is a psychostimulant that causes increased locomotor activity. And so this model is often used as a model for initial drug sensitivity, which has been has a relationship with drug taking behavior in humans as well. This is just an example of 2 different inbred mouse strains that differ for this phenotype. You see day 1, day 2, and day 3, and this is the distance moved in this open field. We also do a more repeated exposures, something called behavioral or psychomotor sensitization, where the animals are given repeated doses of cocaine and their locomotor activity increases upon repeated exposure, even to the same dose. And this is thought to reflect underlying neural adaptations that are occurring in response to chronic exposure to the drug. So we do this kind of all within the framework of a larger project, which is called the CSNA, and that is centered at the Jackson Laboratory in Bar Harbor. It's a big program project of which we're one project here, project 2. We work with collaborators from the University of Binghamton and also the Jackson Laboratory. And basically what we're doing is collecting multiple addiction-related phenotypes on all of these collaborative cross strains, as well as their founders, and another related strain called the diversity outbred. We're looking at doing genetic analysis, looking at things like heritability, genetic mapping. We're looking at gene expression across all of these strains and hoping to identify some neurobiological mechanisms of addiction and create some new addiction models that can be used in the addiction research community. So in my lab, opportunities include animal work. Obviously, we do a lot of behavioral testing with mice, bench experiments. We do a lot of, DNA experiments, genotyping, qPCR, sequencing. There's availability of kind of more data analysis projects or computational studies to look at genetic mapping for the different behaviors that we're measuring. And we've also recently kind of started to extend our work into microbiome studies with a collaborator here on campus, Ian Carroll, that works in the CGI BD. So if you're interested, you can contact me at my email here. We're happy to take students for course credit, honors thesis, volunteer. Occasionally, we have students that work for pay, depending on availability of funds at any given time. So come talk to me in the breakout room if you're interested.
Thank you very much. Okay, let's see. I am going to put that one slide back up that has everyone, the numbers there. Yes, I think that this session was a really good example of the wide breadth of different psychology and neuroscience related research that we have going on all across campus. And so if anything there was interesting to you, you want to ask more questions or just introduce yourself to some of the researchers, we'll open again 5 breakout rooms now, and you can see the numbers here. Can everybody see the list? Did I share my screen correctly? No? Yes? Okay. Yeah. Cool. And so researchers, you can see your number there. So you'll join that breakout room when I open them, and everyone else can just go to a breakout room. We'll stay there for about 10 minutes. I'll close them and then you'll have the opportunity to go to another one if you want. If you're able to jump around after you have your questions answered, that's okay with me also. But I'll kind of cut it off at 10 and then we'll go back to another one. Let's see. And if you don't have the latest version of Zoom and you need me to put you into a breakout room, I'm happy to do that. Feel free to put what number you want to go into in the chat and I can put you there. Okay.